All right. Hi, everybody. Let me just get everything up and running. A little late in because I was just double checking that the alignment would work the way it was supposed to. Okay, so I do believe that we finished up on Thursday with this question or part of the assignment. Yeah, that's right. So essentially what we did on week ago on Tuesday was really the questions up to and including question 8. Then on Thursday, we did a lot of work on the Eugene stuff, particularly the electropherograms and the mapping of the sequence reads to the reference. So basically figuring out where our sequence reads were, like which bit of the gene or the DNA that we had actually sequenced. And then today we're going to do a multiple sequence alignment. And that's really based similar to mapping the sequence reads, but here we're actually trying to line everything up perfectly based on how well it matches our reference sequence. Before we get moving on that, which is relatively straightforward, famous last words, right, Eugene? Does anybody have any questions or stuff that they want to go over from the other labs in this sequence? Any issues, or hopefully all of the teething troubles with Eugene have been ironed out. But even if not, then we can fix those too. Anybody have any questions or clarifications, or how do you do this type stuff? Before we get into goal 5.
Okay, so I don't know if I'm doing this right. I can't remember which step it is because I haven't closed it ever since then because I'm scared to.
Yeah, me neither.
Uh, but I'm at the part where it has, I guess, the coding for the yellow, and it has a whole bunch of coding sequences with different colors.
Have you mapped the sequence reads? That sounds like you've already done that step.
I'm not sure.
Okay. So,
And I don't even know if I'm doing this correct.
that's okay. So does it look something like this? You can see what I'm showing you.
No.
No?
Mm-mm.
Hmm.
It's just the three primary colors, like a turquoise, a cyan blue, and a green. And it's nothing but like the different sequences.
Look like this?
No.
So it's not a chromatogram.
Yeah. It doesn't. I have one that is like that, like the ones that you just showed me.
Mhm.
And then I think we had to do another one, but I'm not sure what to call this one.
Hang on a second. So let's go back here This is again mapping the sequence reads. And we've got that. Let's add a few more. Sequences. It'll take a little while. Do. God damn it, you pain in the very butt. All right, let's try something different this time.
If you go to the participants in the chat, and I think it's you click on more, you can give us access to share our screens.
Oh yeah, give me a sec. Let's just first see if let's do that while that's more. Now it bloody works. How do I do that? Is that in security? Oh, there you go. Sweet. All right, before you share your screen, because I'll need to stop sharing mine. Is this what it looks like?
No.
Oh, okay. So share your screen and show me what you've got. You should be able to do that now. Yeah. Cool. I'm glad I've learned how to do that. It's the first time I've done it. Oh, right. Now I know what you're looking at. So you're looking at the reference sequence, and the different colors represent different parts of the sequence. So that kind of weird green color in the middle is the genomic DNA part of the sequence. So that's what you'll see in the DNA of the fly. The kind of grass green kind of color, I guess, that is the what will be transcribed. So that's telling you that there's an intron between the two. And then the light bluish weird color is the coding sequence, what will be translated into protein.
So referring back to the Word document, which question would this be pertaining to? I can go ahead and stop sharing my screen.
Sure. So that's really kind of question 8 and before. Yeah, question 7, question 8, I think are really what those refer to. So shame we can't just like pick which question you want to go to. Yeah, so you'll use that diagram to answer this question about the actual gene itself. And then like put some numbers to those features.
Okay. Yeah, and I think also what got me tripped up was number 10.
Okay, so actually share the screen with me again. and I'll be able to
Mhm.
see what you've done. Okay, so you haven't mapped your sequences yet. So go up to Tools at the top, click on it,
Okay.
drag down to Sanger Data Analysis, the one at the top. Go to there, Map Reads to Reference. Yeah, so
Because I had tried doing this once with this particular one,
Mhm.
but it gave me a red big no.
Okay, so let's give it another shot just in case, because you never know.
Okay.
Go down to the Add button and find the sequence,
The file
sequence
called
files.
yellow.
No. Click on that, or is that the file? Oh, okay, right. So you need to download the good yellow sequences zip folder from Blackboard, unless you've done that already.
Yes. The good yellow sequence.
That's it. So double click on that.
Oh, but that's the unzipped file.
That's the zip file. Just double click on it. I'll walk you through it.
Okay.
Oh, yeah, I'm sorry. You were right. Remove that. You're going to have to select it first. So go to your desktop.
Oh, okay. All right. Can you see it?
No, yeah, you need to go to your desktop or your Windows File Explorer or whatever you
Okay.
use to actually look at files on your computer.
So I'm at my good yellow sequences and I have all my files.
I can't see what you're looking at. I
Oh,
can only see Eugene. If you want me to
um,
see what you're looking at, you have to share your desktop, not just Eugene.
oh, okay. Let me try this again. Okay, can you see it?
Oh yeah. Okay. So oh, you've extracted that already?
Yes.
Oh, fine. Um, it's easier to keep them all in the same file, but that doesn't really matter. So go back to Eugene and add, and now go to your desktop.
Mm.
There you go. And select a bunch of those. Like,
Like 3.
yeah, I wouldn't use the FA, the sequence FA ones. Get rid of
Okay.
those and then just, yeah, select 3 or 4 of the other ones. There you
Okay.
go. Open. And now go down and click map, which, fingers crossed,
Yeah.
will work. It's always a little nerve-wracking because it often doesn't tell you it didn't work until right at the very end. Hey, it did work. The issue there that I discovered last class or lab is that this map sequence read really hates sequences that you've modified yourself, which is kind of
Mm,
weird. So if you kind of scroll to the right, keep going, there you go. You see now where your sequence maps to, right? So this will allow you to answer the next few questions, like what bits of the gene does your sequence read cover, for example. So the numbers at the top, right, where it says like 115 to 168 on your
mm,
window, right, that's the basis of the reference sequence, right? And so you now need to go back and look at your reference sequence, right? If you click on yellow again, right, that's like 102. All right, so if you start at 102, which is
mm.
Okay, that's about where your read starts. Right, so the question is, what does that correspond to? Right, that looks like it's mRNA, right? What part of the mRNA is it?
Does it tell me like right here?
No, you're going to have to refer back to a previous question that you did, where you worked out the different features of the gene and what their base pairs correspond to.
Okay. Yeah, because I was like, where are they getting all these numbers from?
So for example, we know that base 172 is the first base of the start codon, right? Which you can see, you got rid of the CDS for some reason.
Yeah, that's also another question. I know it's like all the way we were asked about it in the beginning, but like some things just haven't been popping up, especially like that CDS stuff.
Okay, CDS is codon sequence. That's the bit from the start codon to the stop codon.
Hmm.
Okay, so the yellow sequence is actually the mutant sequence. And so base 172 should be a C. So just above and to the left, that's it. Not that one, the one to the right. That's an A. The C next to that, to the right.
Oh,
That
right
one.
here.
Yep. That's the first base of the coding sequence. That should be an A. Right, it should be an ATG because that's the start codon. Does that make sense?
So where the C is and T is, it should be TG.
It should be ATG, but this is the mutant sequence, so it's CTG. Remember that A to C mutation is what the Y1 allele is. That's
Okay.
why it's yellow, right? Because you don't get a protein.
Okay.
No start codon, no translation.
Okay. Yeah, and then I'm assuming I'm still behind from
Yeah, you
what
are a
we're
bit,
going over
but that's
today.
fine.
Okay.
So that will help you answer those questions.
Hmm.
The mapped read, so if you go back over to your object window, which is the one on the top left, and click on yellow Sanger read, keep going down, that one. Double click on that. Right, this will allow you to answer the questions, what is it, 9 to 14? Yeah, 9 to 14.
Okay. And then I'll be caught up from then.
Mhm.
Okay. All right. Thank you.
No worries. So again, you always just have to go back to like what is the nature of the mutation? Where is that change? What's the consequence of that mutation? Right? What phenotype do you see in homozygous recessive individuals? Keep thinking about that stuff, then these questions will be relatively straightforward to answer. Does anybody else have any questions about particularly the mapping sequence reads? I know it was a little confusing last week because we had all kinds of issues, but okay, so to finish up, what we're going to do now is a multiple sequence alignment. And so this is a kind of serves two purposes. One is it's a really nice visual way of looking to see how these things line up, right? So in this case, I know. In this case, what we're looking for is what changes between our mutant and our wild type sequences, right? That's the purpose of this alignment. There's a package out there, love. All right. And that's a really neat way of visualizing it. Other times, what I've used this for a lot is if you have genes from the same gene from related species, or somewhat related species, you can actually use this to show how do these things, what bits are similar and what bits are different, right? And typically the bits that are similar or the same are the ones that are important, right? Because they're shared amongst all versions of that. And then going on from that, if you wish to do so, you can actually use these alignments to build phylogenetic trees, right? So trees showing the evolutionary relationship between species, right? And so that's kind of like the series of steps. I have actually done that before. It's not really my specialty. Other people here, like Dr. Border, for example, does this in her sleep, a matter of routine, for the kind of work that she does, which is a lot of taxonomy and phylogenetics and stuff. You won't mess anything up, Adam. It's all pretty straightforward. Okay, so the way we're going to do this is famous last words. You need to open a bunch of sequence reads, right? And so this is kind of messy down here, but you can see there's a whole bunch of these sequence reads down here, right? And the easiest way to do that is just to go to open, go to your file with the sequences that you want, wherever that might be, and then just Control, or you can even do Shift, and select a bunch, like 6 there. Okay, and then just click Open. And these will all now open up down here. Now, there's 2 steps to this. One, we've got to group all these things together in like a project, right? And then the second is we'll align them all. So the first thing to do is group them together. And the way to do that is to click on one, press down Shift on your keyboard, and then select those. Probably Control is better, actually. Yeah. Hold down Control. There you go. Right, so I've selected the sequence reads that I'm interested in, and now still holding down Control. Where's our yellow sequence? It's in there somewhere. It's just called a sequence. Select that one too. Right, so that's telling the program that you want to do this project with our reference sequence, whatever you've called that. I know Adam called his yellow because that's what you're told to do. I called mine sequence because I'm too lazy to change it. And you should have a whole bunch of sequence reads, and you're telling the program to group them together. Now, before taking your finger off the Control button, if I could show you, it's still there. There you go. How cool is that? Right click on that one. Right, that will bring up another menu. Now you go to Export Import and go across and export sequences as alignment. Okay, that'll bring up this box. You just go export. You don't bother doing anything funky. Now that looks like absolute complete junk, right? This isn't telling you any useful information at all. The one down at the bottom, I wonder if I can yeah, that's neater. Let's put that up there. You can rearrange these if you want. I'd put the reference sequence up the top just because it makes it a little easier to visualize, I find. Now, we want to get rid of these ends because these ends will interfere with the alignment. Right, as you can see, they all kind of share stuff, right, but they're not aligned yet. Oops, wrong way. Go back this way. Okay, so let's get rid of these because these are just going to mess things up. Let's go to the beginning of the long sequence of T's. Right, so you select those. So you put your select one, hold down Shift, select the rest, right click, edit, and then remove selection. What you're doing is you're just tidying things up. It means it's a lot easier for the computer or the program to line these things together because it doesn't have to deal with these weird bits of sequence that, you know, don't really help. Right, so just do that for all of them. Make a nice pretty alignment. Yeah, no, I don't want to do that. Go away. I don't know what's going on with this one. I might just delete that one entirely, to be honest. There you go. Don't have to worry about it then. And same here. Now, it's not going to hurt to have a look over the other end of the sequence, but that all looks pretty fine to me. Typically, you don't tend to get into issues with sequence reads at the 3' end until you've gone 600 to 800 base pairs. And ours is only 400, so that's pretty straightforward. Most of the issues tends to be at the 5' end at the beginning of the sequence read. Everybody with me so far? Give you time to catch up. This is, believe it or not, actually one of the simpler things to do in Eugene. As I've said, this is an astonishingly powerful program. So your alignment should be pretty much the same. What's going to differ is the sequences you use to do the alignment. So you can pick whichever ones you want. If there's anything obviously wonky there, you can take it out. But it shouldn't make any difference which ones you've selected. The only thing that's going to be different is whether they have an A or a C at that base 172. That's the only difference, or should be. It obviously depends which ones you pick as to which ones will have the A or the C. Okay, everybody up, still where we're at? Caught up, same process? Correct. That is 100% correct, Adam. So that's a good point. Let me get back to that point. Okay, so that's the questions. Let's go back to Eugene and actually do this. Okay, so now you need to hold down Shift, select all of those, right? So hold down Shift, click on the first one, and click on the last one. It will select all of those sequences. And now right-click and go down to Align, and align with Cluster W. Don't worry about all this stuff. This is basically the parameters that the program uses to do the alignment. And then just click Align. And in a little while, depending on how new your computer is or what other stuff you have going on, we will twiddle our thumbs. Let sleeping dogs lie. There we go. Okay, so again, we've got the reference sequence at the top, right? These dashes are simply spaces. As of now, those spaces are because there's no sequence there. We'll see if there's any other difference. So we kind of scroll along, and now we hit the first kind of good sequence, right? And what you can see at the top, right, we've got both a consensus and we've got a series of bars, right? And so what that tells you is how many, the consensus sequence is the sequence that reflects the majority, right? Essentially what is the base that is most common amongst all of the sequences that are lined up. The bars show how big of the proportion that consensus is represented by. Pardon me. So if you have here, for example, 100%, oh, hang on a second, 100% of the bases at that position are T's. If you look here, only 83% are T's. One of them is a gap, and the gap exists because there's a space here for this sequence. It was missing from the sequence read, right? When we chopped it off when we cleaned things up. Okay, before we get any further, Adam, show me what you've got, and I'll see if I can figure out what you need to do. This screen sharing deal is really cool. I feel like I'm a remote technician. Oh, that's fine. So what you've done is the first step. Remember, there are two steps. Now, hold down Shift and click on the top one.
Okay.
And then still holding down Shift, click on the bottom one. Not that one. All of them.
Not. Oh, okay.
Right. And now right click and go down to Align and select Cluster W, that one. And then just click Align. Now we see whether Adam has a faster laptop than I do. Probably. Mine's pretty old. That's weird.
Yeah, because like I have the top portion that you have.
Yeah, that's at present somewhat meaningless because the sequences aren't actually aligned.
And I was double checking to see like, oh, okay, he has nothing but T's, 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 T's. So let me see if I have that, but
Right.
it
I
didn't
wonder
pop
why
up.
it's not aligning. Okay, so all right, so close that one. Let's open up. Let's start it again. So at the top right where you see that little red box with the X in it?
Mm-hmm.
Just close that and open up some other sequence files from that Good Yellow Sequences folder. I'd open them from, oh yeah, you are. So yeah, take like 4 or 5. There you go. Now scroll down in your objects view and select those. So click Control and then select each one in turn. And then scroll up and find your yellow reference sequence. That's it. Unclick that. You don't want that. Anything else selected up there? Yeah, unselect those.
Let's see, I did 1, 2, 3, 4, 5.
Okay, what's above KRAM? Oh no, oh yeah, get rid of that. That's it. Now right click on one of those blue ones.
Export sequences.
As alignment. And then just click export. All right, so grab that NC one and move it to the top. I would go to the beginning of the sequences of T's when you're editing.
Okay.
Just because those even further to the run of T's.
Oh, okay.
You see what I mean? Yeah, that'll do.
Edit. Move selection. Sorry about all this.
No, not at all. This is actually kind of neat. In some ways, this is better than when I do it in person in lab because I can actually see what you're doing.
Mm-hmm. Okay, so
okay, so now you've edited all of those. Now select all of the ones there. So hold down Shift and yeah, start at the top and then select the NC sequence too. There you go, that'll do. Now right click, align with cluster W, click yay.
Fingers crossed.
It's always a little nerve-wracking because you never know if it's going to spit out an error, but this should work. It may be that there's some other stuff included in your last one which threw things off. Yay! So now what I'd suggest doing is select the NC sequence in the box on the left. Click on that. And then drag that up to the top. Will it let you do that? Select it in the box on the left.
Oh, okay, I don't know what I just did.
Click on it and then see if you can drag it. There you go. And that just makes it a little easier to visualize.
Okay.
All right, so you need to go all the way back over to the left. You're on the tail end of the sequence there.
Yeah.
So at the bottom, you'll see there's a slider box. Just grab that gray box and then just drag it all the way over. And then a little bit back. What is going on with your...
There's nothing.
Oh, that's weird. Okay. All right, close that window out.
There's no undo button.
Doesn't appear so. Go back to your objects box and then scroll down to the bottom. Okay, click on that. Double click on it. What the?
Nothing. But there is some.
Crab sticks. All right. So third time's the charm, Adam. Oh, there we go.
It's towards the very end for some reason.
That's super weird. Well, anyway, keep scrolling till you get to the.
Yeah, that's at the
Not that
very.
way. Go that way.
I don't know. Even
No,
know
that's not right. So that's telling you that your reference sequence is only 1,000 base pairs long. Something's wrong there. Okay, let's try this again.
I can see.
Yeah, your reference sequence isn't the right size though, and your sequence reads don't show up. Okay, so close that one out again. We'll try a third time. Okay, so you can actually right click on that and delete it. There you go. Let's get rid of that. We don't want that anymore. Okay, so do what you did before. Select those 5 sequence files. You want to use Control, not Shift.
Okay. Oops.
Sorry. I'm sure this is very helpful for other people too. That's it. You're good. Keep going up. You need to then select that yellow. Right click.
Export sequences as alignment.
Yep. You're gonna be a pro at this by the time you finish.
No.
Okay, now before anything else, I think this was me actually screwing you up by trying to be fancy. Select the NC sequence on the left, and then drag it up to the top. Okay, now do the editing. Shouldn't take you very long.
Okay. Oops.
So hopefully this will be benefiting others because you all have to do essentially the same thing.
Okay, here we go. We're almost there.
I have my fingers and my toes crossed, Adam. It's quite uncomfortable.
Okay.
All right, Shift, select all.
Oops.
Okay, right click, align with cluster W. Fingers crossed. I think it was me getting you to move around the reference sequence that caused the problems. I think that was my fault, not yours. I think that caused the alignment to reset itself or something funky. Okay, so yeah, this looks a lot better. Okay, don't change anything there. Don't touch it.
Okay.
Leave it
You
alone.
want to do this? You can do it. I don't want to touch it.
Okay, so grab that gray box down at the bottom, or you can even use the slider down, down, down. Either that one, or you see at the bottom where that's that dark gray line with a bigger, thicker bit. You can also grab that too. All right, so now you're seeing, I don't know why your consensus bars are in plaid. I think that's a little bit sad. Mine's just normal gray.
I have no
But anyway,
idea.
special Scottish consensus bars. I don't know.
Okay.
All right, so now essentially this is all of your sequences lining up. And as you can see, other than some like weirdness at the beginning, they line up really nicely. So keep scrolling to the right. Stop. Okay, so where on there, you can actually look at the bars at the top. Where's there a difference?
172 base pair,
And what does that base correspond to?
CA.
Okay, but what is that? Why is there a difference there? What have we been
Because we
looking
have
at?
is it because we have a mismatch?
That's correct. So there's a difference between your reference sequence and what, 3 of your sequence
Codon.
reads. But what is that ATG or CTG?
That's our what do you call that sequence? That's where the mutation, not the mutation, the protein is missing.
Now that's the mutation, right? So that's the C is the mutation. It should be ATG. What is ATG? What does that code for in a protein?
The adenine, thymine, and guanine.
No, that's DNA. What does the codon code for? The ATG or AUG codon. What does that code for?
UAG.
Yes. But what does that code for? Like what amino acid?
Um, Methyl.
Methionine. That's right. It's the start codon. So that's
Mhm.
the very first amino acid of all proteins, at least as they're translated. So if you don't have a codon for methionine there, are you going to get protein?
No.
No, because you don't have a start codon, right? So that's why your flies are yellow, that have that single base change. And so this shows you graphically, both in the actual alignment, the colored blocks, but also in the consensus bars at the top. It shows you this is the thing you're interested in, right? This is the base change that makes the difference between brown-bodied versus yellow-bodied flies. It's all just that one base.
Okay.
Ta-da!
Yeah. Woo.
Yeah, awesome. So yeah, the last time it didn't work, that was my fault. Sorry about that. So yeah, once you've done the alignment, don't touch it.
Okay.
That's the lesson there. And now if you kind of scroll to the right further, you'll see there really shouldn't be any other differences, right? It's all the same. The only thing that changes, so you see towards the end, right?
Mhm.
Those ends are
The three
basic.
prime.
Yeah, the 3' and the sequence reads. Those ends are bases that couldn't be called, so we don't know what base they are according to the sequence machine. And the dashes are gaps that are introduced so that things line up. So basically what that's telling you is that in those sequence reads, there's like an extra letter or two, which causes the whole alignment to be kind of stretched a bit so it all lines up.
Oh, okay. Cool. Science is cool.
It's pretty neat. And it's
All right.
really cool so you can do this for proteins as well. Actually, it's probably even more informative for proteins if you're looking at comparisons between species, because DNA can change a lot, protein changes less. And then with proteins, you can actually see which regions of the protein are conserved, right? Presumably they do something important.
Okay. All right.
So
Thank you.
no worries. Good job.
Hopefully with I can do all this first and then answer the questions.
Yeah, absolutely.
Okay.
So,
I don't have to
uh,
go by it like one by one by one.
well, if you have that up and open and then you go and read through the questions, question 14 is all about those consensus bars at the top,
Mhm.
right? So you kind of know what uh, those mean, right? Question 15 is really asking the same question, but in a different way, right? Again, about the consensus bars.
Okay.
Question 16 is about the nucleotide that you care about the most. Wink, wink, nudge, nudge.
And then that would be where that protein or mutation is.
Well, I mean, that would be me telling you the answer, wouldn't it now, Adam? Not
Oh
going to
yeah,
do that.
sorry.
But most likely it's present on your screen right now.
Oh, all right. Let
Uh-huh.
me change it then. I'm kidding.
Question 17 is all about those dashes and what they mean.
And you had said that they were the codons that didn't have a match, right? Or there were spaces to which they couldn't match.
Yeah, I actually have to change this. The person that wrote this didn't quite put in exactly what they mean. So I'll edit that in a actually I can get back to that in a second. Question 18 is really all about like the whole lab, so what is it that you've been working on, right? You've been uh working on, the genetic basis between point mutation and like the phenotype, body color of a fly. You've been figuring out why that genetic change led to that particular phenotype, what kind of mutation that is, um, the mechanism by which that mutation leads to, uh, the yellow phenotype, and whether or not that's a big deal, basically, and that's really question 18. But let me just quickly, um,
So in the end, that's basically it for all the Eugene stuff.
that's correct. I'm just gonna, while we're talking, modify. I've modified it in for a different semester, but um, this one was from last summer, so I don't think that one was changed. Give me just a moment, because I don't want there to be any confusion about the answer. Is that the dog, Evie? He's stop it digging into the couch. Honestly. There you go. That's a better way of wording things, I think.
All right, I'll be sure to check that out later. Thank you.
That's pretty much it. If you want to stop sharing your screen. All right. So question is, anybody else have, wouldn't say issues, that's not quite the right word, need help working through this? That's a better way of putting it, I think. Cool. All right, everybody. So this isn't due until, what was it, next Tuesday?
It's
Oh,
this
this
Thursday.
Thursday. This Thursday. That's right. Yeah, thank you, Adam. Trying to remember off the top of my head. So if you have any questions about it, like still can't figure out, what's going on and why it doesn't look the way it's supposed to, give me a shout and I'll be more than happy to help out. Even do some like funky, remote access type stuff like we did today. Should be neat. Otherwise, thanks very much everybody for your time and attention. It's been fun, interesting. I learn every time. I learn something about Eugene, even if I might not always want to. But yeah, I hope you really enjoyed this lab, even with its online ness, which is kind of a little odd sometimes. But anyway, I think hopefully you've learned a lot. I think it'll really benefit you in the future. Anyway, That's it from me. Going to say goodbye, and hopefully I'll see you all around on campus in the future. All right. Bye, everybody.
Bye. Thank you. Have a good summer.
No worries. You too.
Bye. Thank you. Have a good summer, Professor.
Thanks. You too.
Thank you. Bye.
And Tripocelli starts playing. I wouldn't try impersonating him. I don't think that would be anything you'd want to watch again. Cool. All right, everybody. Take care.